So it is really my pleasure to be able to introduce to you today Professor MacDonald from the University of Alberta. And we were really fortunate to be able to recruit Patrick as a visiting professor at the University of Southern Denmark with the generous support of the Danish Diabetes Academy. And although this current situation is making travel impossible, I would like to say that the visiting professorship has already strengthened the collaboration between Patrick's and my group. So a bit about Patrick. Patrick received his PhD back in 2003 in physiology, where he studied voltage-dependent potassium challenge at the University of Toronto. Afterwards, he continued to study electrophysical mechanisms and properties of islet cell exocytosis as a postdoctoral fellow first in Lund and afterwards at Oxford University. Then in 2006, he was recruited back to Canada, where he has led an extraordinary line of research into islet exocytosis. He has so far more than 100 publications and 8,000 citations. This has earned him several positions. So he's now the director of the Alberta Diabetes Institute Islet Core, and he's also a professor of pharmacology. In addition to this, he has earned several awards, such as the Piavsky Young Investigator Award, and recently he has also been named as a Canada Research Chair. So I think it's really clear that Patrick is a real powerhouse in islet cell research, and that we're very fortunate to have him associated with Danish research. So without further, Patrick, the virtual floor is yours.
Great, thank you so much, Jesper. Let's see, I'll pull up should be able to share this, I think. There we go. Yeah, it's it's a real pleasure to be here. Of course, I hoped and was very excited to be spending some time in Denmark now and working more closely with Jesper and others. But like he said, we've managed to continue on improving collaboration. I'll point out that we've also started some interesting stuff with other investigators in Denmark as well. So I'd really like to thank the Danish Diabetes Academy for having me, and I look forward to continued collaboration whenever and travel whenever that might be possible. Hopefully it's not in the too distant future. So I'm going to talk about some of our recent work and some of the stuff that's led to collaboration with Jesper, looking at islet cell subtypes in human islets in health and diabetes. I'm going to talk mostly about some unpublished stuff on alpha cells, and given the audience that we have here, I'm really hoping for some feedback on the work that we're doing. I'd really like to hear your thoughts on it, because there are still several things we're trying to figure out about how alpha cells work and what we're seeing in our data. Given the inability to travel at this time, I know people are keen to see strange and distant lands. So here's a few pictures from Edmonton. I'll admit I did not take all of these. Some of these are from Google searches, but they're all of Edmonton and pretty indicative actually of what things are like here right now. So this is where I am. We work on islet hormone secretion, as Jesper mentioned, insulin and glucagon. And all I really want to point out in this introduction is something you know already, that insulin secretion is impaired in people with type 2 diabetes. This is showing a reduction in vivo insulin responses during the progression from normal glucose tolerance to type 2 diabetes. But what I want to point out is that this impairment in insulin secretory function can be seen at the level of the pancreas, isolated pancreatic islets, and even at the level of single pancreatic beta cells. So an impairment in insulin secretion in type 2 diabetes can be seen all the way down to individual pancreatic beta cells. And the same is true with glucagon, where we see dysregulation of glucagon secretion all the way down to the level of the single pancreatic alpha cell. And this is a nice recent paper from Sebastian Barg's group in Uppsala. And so these are the kinds of things that we're interested in studying, really interested in trying to figure out what's going on at the level of the islet and the single cell with respect to insulin and glucagon secretion and mechanisms for dysfunction. To do this in humans, sometimes I give talks like this. I feel like I'm a bit of a salesperson, but this is a program that I think we're quite proud of here. But to look at islets and islet cells from humans with and without diabetes, we run a human islet isolation program. And this is, I think, a little bit unique in the sense that it's a research-only program we run separately, but in parallel to the clinical transplant program here in Edmonton. And I think we're probably the only islet cell electrophysiology group that isolates its own human islets. So if you want more information on this, there's a couple of websites you can check out here or here. You can actually pull up some information on some data and information on the preps that we do in Edmonton. This has grown quite a lot just with respect to the Danish Diabetes Academy. This award visiting professorship has spurred interactions through this islet core program with other groups in Denmark already, and those are being set up now. But what I'm going to talk about is work that follows on from a paper that we published earlier this year describing something called pancreas patch-seq. And what that is we take islet cells from these human donors, and Xiaqing Dai in the lab runs electrophysiologic assays on these cells, alpha, beta, and other islet cell types. And then we collect them and then send them to our collaborator at Stanford, Joan Komunis, who's in the group of Stephen Quake. And he does single-cell transcriptomics on these. So we're collecting detailed electrophysiologic profiles, looking at ion channel activity, membrane fusion, exocytosis, and things like that, and sequencing the cells. So now we can connect these functional properties, these membrane function properties, to transcriptomic data. In that paper, and I'm not going to talk a lot about that paper itself, but in that paper, we looked at correlates between gene expression and beta cell exocytosis. This is a non-diabetic beta cell with several positive, green, negative correlates in red of insulin exocytosis, a lot of things that make sense, and you can go into that data and you can mine stuff there looking at potential pathways that regulate beta cell function. One of the interesting things that we can do with that kind of data is look at beta cell heterogeneity in these single cells. We know this is a nice figure from a review by David Hodson showing different markers that are used to identify beta cell subpopulations. I'm not going to go through them all here, but this one here, this RBP4, was one of the first transcriptomic subpopulation markers in the paper by Segerstolpe. And we can show, for example, that RBP4 high and RBP4 low beta cells have different electrophysiologic properties, particularly with respect to their sodium channel activities. And then in some unpublished work by Bikram Roy in the lab, he's gone on to show that these subpopulations of beta cells indeed have differential excitability. So the RBP4 negative cells are actually more excitable than the RBP4 positive beta cells. So one of the things we're hoping to do with Jesper, because Jesper has an interest in beta cell subtypes and how they change in type 2 diabetes, we really want to take some of this data that we have and are still generating and apply that to some of the larger data sets that Jesper is working with. Well, what about alpha cells? And what I really want to talk about is work we've been doing for many years now, actually, on alpha cells, and we've started to bring this patch clamp sequencing stuff into that work. Alpha cells are a bit confusing. They behave, differently than beta cells, which is not surprising, I suppose. And Shuching in the lab started looking at how glucose might be controlling alpha cell exocytosis in a cell autonomous manner. So these are single alpha cells. In this case, these are identified by immunostaining, not sequencing. But what she sees is that glucose, as it increases, suppresses alpha cell exocytosis in beta cells. Beta cells will be low at low glucose and then increase, of course. To make a long story short, she showed that this is due to the fact that glucose in alpha cells is regulating a calcium channel at low glucose. So at low glucose, you have a high activity of agatoxin sensitive calcium channel, which is the PQ type calcium channel, not the L type calcium channel that you might be thinking of in beta cells. Just ignore this. We use barium in these experiments because it's easier to measure than calcium currents. But that doesn't really matter. And if you look at exocytosis in alpha cells under that low glucose condition, you can see that it's controlled by these agatoxin sensitive PQ channels, not the isradipine sensitive L type channels. This is consistent really with what others have shown, actually, particularly from Patrick Roersman's group, showing that alpha cell exocytosis is linked to PQ type channels. And what we've brought into that story here is that glucose suppression of the PQ type calcium channels leads to suppression of that exocytotic response. So before we go too much further, I just want to take a step back and say, I know that physiologically alpha cell glucagon secretion a number of different factors, and we're not really trying to say here that the intrinsic alpha cell is responsible for the physiologic changes necessarily in glucagon secretion. What I think our data is saying is that the glucose levels that the alpha cell is seeing are going to modulate the alpha cell responses to some of these other signals by controlling the calcium channel activity. Okay, so I hope that makes sense. In a way, I think alpha cells are really a fantastic integrator of many signals. So we're really just talking about the cell phenotypes of these cells and how that might dictate how they would respond to other signals. So we have our glucose suppression of alpha cell exocytosis on the left here. And if we bring that, the transcriptomics into it, we can start to correlate gene expression in individual alpha cells to their exocytotic responses under different glucose concentrations. So we're looking at 1 glucose and 10 glucose here, consistent with that suppression of exocytosis. We get a leftward shift in these correlations as we increase glucose. And then we can go into this transcriptomic data and look for pathways that are either driving this relationship to the left or to the right at 10 and 1 glucose respectively. And we can look at pathways that, we can see some pathways that might be contributing to the glucose regulation or glucose modulation of alpha cell exocytosis. One thing that jumps up here is pathways involved in mitochondrial respiratory chain complex assembly. It's really quite interesting because this pathway is a positive correlate of alpha cell exocytosis under low glucose conditions and a negative correlate of exocytosis under high glucose conditions. So at low glucose, it's pushing this distribution to the right, and at high glucose, it's pushing that distribution to the left. You can see a number of these transcripts involved in this pathway, many of them in complex 1, flip their correlations as you play with glucose concentration. We can show the same thing essentially in mouse alpha cells, and I'm just showing this on the right here to show we've looked at the requirements for glucose metabolism per se in what we're seeing. The other thing that looked sort of interesting in this pathway, or this group of pathways, was this one here, endocrine system development. And we see that this pathway is a negative correlate of alpha cell exocytosis at low glucose. So what that means is that these transcripts are popping up in alpha cells that are responding poorly under low glucose conditions, so suggesting maybe there's something going on with developmental state in terms of the responsiveness of these alpha cells to depolarization-induced exocytosis. If we look now at type 2 diabetes, what we see is that alpha cells are dysfunctional, which is, I think, not surprising. They're low responding at low glucose, and if anything, they increase with increasing glucose. I think it's quite interesting that these correlations, the relationship to glucose and the distribution of these correlations, the transcript correlations, flip-flop in type 2 diabetes. So now high glucose is driving these correlations to the right. And when we look for pathways that might be driving, a whole bunch of things pop up related to development and cell fate. So we have cell fate, pancreas development. Some of the contributors to that are shown on the bottom right here. And I mean, this starts to get quite far away from my area of expertise, to be honest, because I'm not a pancreas development person. But a number of these look very familiar in terms of islet cell lineage. NeuroD, for example, PAX6, and so on. And so, you know, it fits with, and I don't think this is really our idea, but it fits with the idea that we may be having some sort of impaired identity in alpha cells in type 2 diabetes. This is a nice figure from a review by Mark Husing's group. There is, of course, evidence that alpha cells can be plastic, depending on the conditions in which you find them. This is from Pedro Herrera, showing extreme loss of beta cells leading to alpha cell conversion into beta cells. Klaus Kestner's published a few years ago now that alpha cells may be epigenetically more plastic than beta cells. And so maybe some of what we're seeing in terms of the changing alpha cell phenotypes is related to alpha cell plasticity. He published this nice paper this year showing that a number of gene sets linked to immaturity in alpha cells are actually upregulated in type 2 diabetes in alpha cells. So there's this idea that alpha cells may be somehow in a less mature state generally and more plastic than beta cells. We worked with Sung Kim in Stanford a few years ago on his model. So the question is, are these changes in electrical phenotypes we're seeing in alpha cells, can they in any way be related to the altered differentiation state of alpha cells? So we worked with Sung Kim a few years ago on his model of alpha cell transdifferentiation, where he's knocking out in alpha cells ARX and DNMT methyltransferase. He shows that I think it's something like 40% or so over 8 or so weeks turn alpha cells lineage traced turn into beta cells. And in that study, we did some electrophysiology looking at exocytosis. So this is an exocytosis measurement in an alpha cell, which is not converted, so we know it's not changed into a beta cell. And again, we see this glucose-dependent suppression of alpha cell exocytosis. And if you look at cells which were previously alpha cells and now converted to cells, these guys show up as yellow here, we get a flip-flop in that glucose relationship between glucose and facilitation of exocytosis. So now increasing glucose, increase exocytosis. There's another measure that's going to introduce here, which is about how the sodium channels behave in alpha cells versus beta cells. Several years ago, I think it was Sven Gopal, Sebastian Barg in Patrick Rorsman's group showed that alpha cells and beta cells in mice can be distinguished by their characteristic inactivation properties of this sodium channel. So if any electrophysiologists are listening, they already know this. In mouse cells and in rats too, alpha cells and beta cells can be quite easily distinguished by the inactivation properties of their sodium channels, right? So beta cells, alpha cells to the right, beta cells to the left. And in these converted alpha cells, you see we shift this sodium channel inactivation to the left towards a beta cell phenotype. And this happens in concert with upregulation of that beta cell sodium channel transcript, SCN9A. If we stick to that now, looking at this way in which electrophysiologists will identify the alpha and beta cells, and we do this in high-fat fed mice, where we have identified all these alpha cells by glucagon immunostaining, we see something that looks pretty similar. So a leftward shift. Again, these are all alpha cells, but now we have a population of alpha cells displaying a beta cell-like sodium channel inactivation. And that so we're taking in the middle here the half and half action, so this halfway point voltage for an activation. You see alpha cells shifting to more negative potentials, and that's a beta cell phenotype. In concert with that, we see upregulation in the peptide alpha cells of SCN9A, that beta cell sodium channel. And in the high-fat alpha cells from mice, if you look at the chow-fed, first of all, we see exactly the same thing we saw in the humans: suppression of exocytosis by the toxin, but not the L-type channel blocker, it's ratapine. In the high-fat diet animals, or alpha cells, we see no effect of channel blocker, and suppression by the L-type channel blocker is ratapine. Again, that's a beta cell-like phenotype. These are not beta cells because they're all glucagon expressing. Going back to the human data, and we're coming towards, well, not quite to the end yet, a little more to show. The problem with using that as an electrophysiologic identifier of cell types in humans is that the sodium channels simply can't be used in that way. And that's illustrated here, where you can't separate, if you have a voltage-dependent inactivation of minus 50, that doesn't tell you if you're looking at an alpha cell or beta cell because the distributions overlap so much. We can look at the sodium channel at the alpha cells, of course, because it's still quite important, and I'm not going to go into that work per se, but I'll highlight that the sodium channels correlate with a lot of things that we know are very important for alpha cell function. So here's the alpha cell, these are non-diabetic alpha cells. These are the alpha cell sodium channels, the PQ type calcium channels, and other transcription factors and things known to be important in alpha cells. But we can't use this measure to separate electrically alpha and beta cells. What we can do is try to figure out a way in which we can use our electrophysiologic data, which has much more than just alpha or sodium currents and exocytosis. We're looking at calcium currents. We're looking at different properties of the inactivation and activation of these channels. We're looking at early and late exocytotic responses and so on. We don't need to really go through all of this. But we have, for all of these couple of thousand cells now, we have, with electrophysiology and sequencing, we have what you might call an electrophysiologic fingerprint. And a few years ago, Linford Bryant in Oxford published this paper, which is actually quite cool, where he's using data like this to distinguish alpha and beta cells in rodents and improve cell type identification there. So without going through how we did it, we worked with Linford to generate some machine learning models, and this is done with a grad student in the lab, Theo De Santos, to basically take several of our electrophysiologic parameters train models to identify alpha and beta cells in our human data. And so what we do with this is effectively generate a single number, which would be a probability with which the cell or the electrophysiologic fingerprint matches an alpha cell. So everything that clusters up near 1.0, the model is telling us should be an alpha cell. Everything that's clustering down towards 0, the model tells us that should be a beta cell. So you can see the difference between distinguishing cell types by sodium channel inactivation and by these fingerprints here. All of this is connected to transcriptomic data, and so we can confirm, for example, that the model works. Right? So alpha cells cluster over here, identified by several canonical markers, beta cells down here. So this is all done in non-diabetic beta and alpha cells, and we can use this information now to look at how I guess you might call it a functional or electrophysiologic identity of the alpha cells change in type 2 diabetes. And we actually, between Linford and Theo, we made models. All of them basically told us the same thing, and that is in type 2 diabetes, alpha cells lose some of their electrophysiologic identity and they shift to the left here. And then we can make correlations between transcriptomics and these alpha cell probabilities to see what pathways might be driving this shift towards the left or away from our mature alpha cell modeling. And several interesting things come up. I've highlighted a few here, ARX and DNMT, because I showed the knockout data from Sung Kim. But what we see is that alpha cells that shift towards the left or lose this alpha cell probability tend to correlate with expression of these markers. DNMT suggesting perhaps again that lineage or maturation state is somehow important. It's very confusing to me because ARX should be an indicator of a mature, happy alpha cell, right? So why are these the ones that appear to be more dysfunctional? We see heterogeneity in the expression of ARX and other markers, and this is really that, in our high expressing ARX alpha cells, these are all alpha cells, by the way, all have the same glucagon essentially, that there are a number of other markers that are high in these cells. Some of them are mature alpha cell markers, some of them are probably markers that are a little more early in the maturation process, like FEV is more of a lineage marker as far as I understand it. The expression of these guys don't really change in type 2 diabetes, but we're interested in trying to figure out whether or not the behavior of these different groups differs. And we correlate now exocytosis at low and high glucose with these markers in non-diabetic alpha cells. We really don't see much of anything. So, the exocytosis in ARX low alpha cells and high alpha cells is pretty much the same in non-diabetic alpha cells. But the picture changes quite a lot in the type 2 diabetic alpha cells, where now we see a number of these markers becoming negative correlates of exocytosis at 1 glucose. So remember, negative correlate means that they're lower than they should be in terms of exocytosis. And I don't think any of these reach a great level of significance, but as a whole, as in the group, we see these group of factors shifting to the right, and the 10 glucose meaning associated with elevated alpha cell exocytosis. And the last piece of data that I'm going to show I think highlights what I'm trying to say, and that is alpha cells that are high in these markers, ISL1 I'm showing, and KX2.2, and ARX and NeuroD, so on. There's no real difference in the non-diabetic cells in how well they undergo exocytosis, but it's these high expressors that have a selective impairment, if you will, in exocytosis at low glucose. So we think that the patch-seq approach is useful. It can be used to define or link functions with transcript expression. I think we'd love to apply this with Jesper to looking at excitability and exocytosis in novel subpopulations of beta cells. And we are continuing to look at glucose regulation of alpha cell function. And we think that there's something going on perhaps with lineage or identity in alpha cell dysfunction in type 2 diabetes. And with that, I'd like to say thanks to all the members in the lab shown on the left here who contributed to this and other work in the lab, collaborators at Stanford involved in the patch-seq stuff, and in Oxford working on some of the modeling. And I didn't really talk about some of this stuff that's related, work with collaborators at Vanderbilt. And of course, we're looking to make use of this data that we're generating and contribute to collaborations throughout Denmark, and particularly with Jesper at STU. So that's all I've got. If there are any questions, I almost forgot this part. You should always thank organ donors and their families when you're working with human islets. It's not an easy thing for people to consent to research use of an organ. And so I think this is very important. Great. Thank you very much. I will stop sharing my screen now if I can. And I'm happy to take any questions. How do I do that? There we go. And I hope I wasn't on mute the whole time.
No, it wasn't, Patrick. Thank you for a great presentation. So there are already questions from the audience. So the first one comes from Agabak. Sorry if I'm not pronouncing it right. The question is it known whether alpha cells in a transient state, that is, that they co-express insulin and glucagon, are actually functional? And the second one, let's start with the first one and then take the second one.
Yeah. Well, the short answer is I don't really know. We do see insulin transcript in some of our alpha cells. It's obviously much lower than beta cells. Insulin transcript, you will find some in I think most cells most islet cells if you look closely enough. But true glucagon insulin co-positive cells, we don't see many of those. And I think if we do, we'd have to be a little careful about how we interpret them and whether they are doublets, for example. We had a little bit of a problem when we started this study with that kind of thing. But the other part of your question about transient states, I think, is interesting because I sort of presented this alpha cell stuff as different populations of alpha cells, but I'm not quite sure that that's necessarily the case. There's this. I have to credit, I think, Jesper with mentioning this to me about beta cells cycling through periods of ER stress, high levels of transcription factors, lower levels of transcription and insulin expression. And so alpha cells may be similar in that way. We have to keep that in mind.
The second question is that the trans or de-differentiation of alpha cells into beta cells has been issued a lot recently. What is the state of knowledge regarding the de-differentiation of beta to alpha cells?
Oh, beta to alpha cells. Well, I guess so. The first part, I think the question was about trans-differentiation of alpha cells and some of the controversy in the literature about some things like GABA, for example, resulting in trans-differentiation, which it seems does not happen. And I'm not really trying to suggest that alpha cells are trans-differentiating into beta cells in type 2 diabetes, but more there's an issue with maturity, perhaps. The beta cell to alpha cell trans-differentiation, I'm not sure I have much to say on that right now. I haven't thought a whole lot about it, to be honest. I mean, there's obviously a lot of work in beta cell de-differentiation. And we haven't really seen much of that in our beta cell data, but we have to go back and look, I think, a little more closely. So a little bit of an unsatisfying answer, I know.
There's another question from, it's difficult with the name, it's Isaric. We have observed that alpha cells exposed to metabolic stress undergo ER stress, but do not undergo apoptosis. This, however, inhibit protein translation. Did you correlate your mRNA?
Yeah, sorry, Jesper, you broke up on me a little bit there, but I do see the question. No, so Decio, the short answer is no, we do not have any protein data with what we're doing. It would be very interesting if we could somehow do that. But what I didn't show is that there are indeed ER stress markers involved. Actually, this morning, before the presentation, I pulled that data out for time. But we do see ER stress markers that correlate with some of the measures we're seeing in type 2 diabetic alpha cells.
And maybe I can ask a question while there isn't anyone. So I was wondering if you have looked into some of this data on differentiation of the pancreas, and if you in that data could trace whether these calcium channels are expressed at different stages of differentiation. Because the switch in channels might represent de-differentiation, or it might represent a directed response.
Yeah, so we looked at, you mentioned calcium channels, and we'd have to look more closely at that, but a differential expression in comparing the non-diabetic and type 2, and actually comparing the chow diet and the high-fat diet alpha cell, really show us anything with respect to calcium channels. So we have that flip in the calcium, the sensitivity of exocytosis to the calcium channel inhibitors, but we do not see changes or nothing obvious in the actual transcript levels of those channels. So it's more about, I think, the regulation and the coupling to the channels than changes in the channels themselves. But I think you're right, though. If we were to really push cells back towards a more undifferentiated state, or really push them towards, another cell type, then we should see some differences.
Thanks. There's another question from Jakob Knudsen. He says, if you have any thoughts on the cause of change in electrical properties of alpha cells in your type 2 diabetic population, is it a change in general expression, or are there other things going on?
Yeah, that's a tricky question, Jakob. So, I've shown correlations with transcription factors, and there's other things that pop up as well. It's very difficult, and I haven't yet been able to pin down what's actually driving some of the changes we see. So why is exocytosis low at 1 glucose in the type 2 diabetic alpha cells? There's no obvious machinery like the channels or snare proteins, snare transcripts that have popped up. Some of them are a little hard to detect, so maybe that has something to do with it. But no, we don't yet know what's actually driving the functional changes. I just see a note in the chat from Decio saying that the sound is pretty bad. Can you hear me okay?
We can hear you, yes.
Okay.
Okay.
If anyone has questions, they are welcome to email me or catch me on Twitter or what have you. So for the poll, can I answer that the scientific content was great? if anyone wants to chat anytime, just feel free to reach out.
That's great.
Yes.
Yeah. But yeah, I don't think there are more questions at the moment.
I think we already taken up a lot of time for this webinar. So thank you, Professor MacDonald, for your presentation and for answering the questions. And thank you, Jesper Lassen, for being the chair of the webinar. And thank you to all of you for attending and for your questions. We really appreciate that. Let's end the webinar. Bye-bye.
Thank you.
Bye, thanks.